Michel Sadelain is our next international laureate that we'll be able to hear from this morning. Michel is the founding director of the newly established Columbia Initiative in Cell Engineering and Cell Therapy at Columbia University in New York. He previously founded and directed the Center for Cell Engineering at Memorial Sloan Kettering Cancer Center. He received his MD from the University of Paris and his PhD from the University of Alberta. Following his postdoctoral research at the Whitehead Institute at MIT, he joined MSK in 1994 and more recently Columbia University in 2024. Michel Sadelain has made several key contributions to the emergence and success of CD19 chimeric antigen receptor, or CAR therapy. His research has contributed to many facets of CAR therapy, including T-cell engineering methodologies via retroviral vectors and gene editing, CAR design, the dual signaling receptor concept, which became known as the second generation CAR, the identification of CD19 as an effective CAR target, the T-cell manufacturing process itself, GMP processes in collaboration with Dr. Isabelle Riviere at MSK, and the successful clinical translation for B-cell malignancies, AML, now expanding into other diseases and other solid tumors. Sadelain is the recipient of the Cancer Research Institute's Coley Award for Distinguished Research in Tumor Immunology, the NYI PLA Inventor of the Year Award, the Pisano, Gabi, Pasteur, Wiseman, Servier, and Leopold Griffuel Awards, the INSERM International Prize, the Breakthrough Prize for Life Sciences. He's also previously served as president of the American Society for Gene and Cell Therapy. The 2024 Canada Gairdner International Awardee, Dr. Michel Sadelain, will be presenting CD19-CAR T cells, Paradigm for the Living Drug Concept. Please join me in welcoming him to the stage.
Thank you. That's not, I need the clock. Close it. Okay. Okay. All right. Thank you very much for the kind introduction, and thank you, first of all, to the Gairdner Foundation for recognizing the work of Zélie Guichard's and my own, and in doing so for recognizing, the value of T-cell engineering. So what I'd like to do today is to tell you the story of the rise of genetically engineered T-cells becoming drugs. First, drugs for cancer therapy, and perhaps soon drugs for other diseases. A few of us some 30, 40 years ago were, of course, interested in advancing immunotherapy to treat cancer, but there was, at the time, quite some disbelief that the immune system could ever succeed in overcoming cancer, and that is because so many of us develop cancer or will develop cancer and succumb to that disease despite having our immune system. Before I get into what led us to want to engineer T cells, I should give you a little bit of background on our immune system. Obviously, our immune system is extraordinarily valuable in keeping us alive and fighting the entry of foreign parasites into our body, may they be viruses or bacteria. And evolution has given us very incredible tools to fight the entry of these parasites, of these undesirable parasites. In what's called the adaptive immune response, we have both T cells that have exquisite specificity to engage molecules that they recognize as not belonging in our body, as well as antibodies, which are produced by B cells. And to build up this line of defense, in the case of T cells, our thymus produces these cells that in blind fashion assemble a set of genes called V genes, D genes, and J genes that produce a collection of T cells that we refer to as a repertoire that is ready in our body to fend off one of those invaders should it come to the body at some time in our lifetime. Some scientists think that we have about 20 million or so of these different T cell receptors in a normal adult. However, in the case of cancer, the invader is not foreign. It comes from us. It is one of our own cells that begins acquiring somatic mutations, and in doing so acquires new properties of invasion, proliferation, failing to die, and immortality. These mutations can sometimes be recognized by the immune system, and in fact the immune system shapes the development of cancer, something that is known as cancer immunoediting. And in some patients, we can use antibodies that will unleash the power of T cells that are formed in some patients that can eliminate these cancers, but in many more, this natural immune response, if you like, is insufficient to prevail over cancer and eliminate the cancer. And for this reason, this led some of us, a handful of us, some 30 years ago or so, to think that we needed a different approach, or at least another approach. And this approach would rely on providing an instruction to the T cells, an instruction to guide the T cell to the cancer, and to go on to kill those cancer cells. And so the field of CARs was born sort of on the fringes of other fields, on the fringes of, on the upper right here, of tumor immunology that has taught us so much about the biology of T cells, the microenvironment, the targets that exist in cancer cells. But it's also different from conventional immunology insofar that these T cells will be repurposed and reprogrammed, and to do this, we need genetic tools. The first genetic tools that were used were retroviral vectors, and today we use more sophisticated tools such as CRISPR to edit the genome of T cells. New receptors had to be created, synthetic receptors that would engage different targets in different ways, and I'll come to that, of course. Those are the molecules that some years ago we named CARs. And these receptors, unlike the physiological T cell receptor that engages what are called HLA peptide complexes, differ in their nature. They have to be on the surface of cells. They can be proteins, glycoproteins, but they could also be a carbohydrate or a glycolipid. And I'll come back to the CD19 target, which is really the poster child, if you like, for this therapy. Because so few were in the scientific world and medical world believed that this approach could ever succeed or be useful, the few of us working on this had to prove it beyond animal studies and bring it to the clinic. And that obliged us to really become cell manufacturers, something that started not in the industry, but in academic centers. And of course, in doing so, the process for making these cells couldn't be that you would use in the research lab, but would have to follow a set of rules set forth in the United States by the FDA. So how to summarize the first 15 or 20 years of this in one slide, I will attempt to do so. When I left the University of Alberta, I wanted to engineer T cells, and there weren't so many places in the world where the rudiments of gene transfer to mammalian cells were being established, and one of those was MIT at the Whitehead Institute. And so I went there to learn the basic principles of genetic engineering at the time. And it took me a few years to come up with this demonstration here that we could take real T cells, primary T cells, in this case from a mouse, and genetically modify them using recombinant retroviruses carrying, in this case, the E. coli LAC-Z gene. This is before even the GFP protein that was mentioned earlier this morning had been discovered. This didn't elicit any interest back then when I presented this for the first time at a World Congress in Budapest. And yet for me and those working with me, this was a magic moment where we thought that we had now a path to go and engineer T cells. And an important decision that we made in retrospect, very critical decision, was that we would study T cell signaling not in what are called leukemic cell lines or hybridomas, as so many others did and taught us many things, but that we would study T cell engineering and T cell signaling in authentic primary cells. And I think it's very largely thanks to that approach, which is admittedly cumbersome and slower than other methods, that we came to uncover molecules that were actually functional in human T cells and ultimately in patients. We had to design new receptors, and here I refer to the cloning of the zeta chain, which is one of multiple chains that T cells use to initiate their activation. 3 groups in 1991 independently cloned this chain and created these fusions receptors with extracellular domains that you see here, because this zeta chain is really an intracellular protein. And in doing so, they found that this zeta chain was sufficient to initiate the activation of a T cell. And then Zelig Eshar, whom I want to name because he's my co-recipient today, introduced this variant on this zeta chain fusion by fusing here elements derived from an immunoglobulin, the heavy and light chains of an antibody, which confer the specificity, the exquisite specificity to different antibodies. And since he's not here, here's a picture of Zelig, whom I last saw in Tel Aviv in February of 2020. But because we could study genes in primary cells and not just in leukemic cells, we realized that these zeta chain fusions were not that effective in redirecting an immune response. You could get a T cell to kill maybe a target once, and then the T cell would collapse in a process known as activation-induced cell death. And so we studied that signaling pathway for quite a while, and eventually realized that if we started combining signaling domains within these molecules, we could now achieve or create, if I may say, T cells that could engage an antigen, kill the targets, the cells that express that antigen, but also go on to proliferate and kill more cells again and proliferate some more and kill again. And this was obviously what we were looking for and the necessary, the obligatory path to come to what we later called a living drug. We needed to identify a target, first for our experimental work, and one that we thought we could eventually bring to the clinic. And we focused on this molecule called CD19. CD19 was known as a molecule found in B cells, cells of the immune system, from which different leukemias and lymphomas occur, B-cell lymphomas and acute lymphoblastic leukemias. And CD19 was not a very popular molecule at the time. It was known to participate in B-cell activation. But we thought that it would be a good target because it is expressed in the majority of these cancers. It is in the body found only in B cells, and even if you destroyed those B cells, well, patients could still fare well. And in 2003, we provided this first proof of concept experiment showing that now you could take human peripheral blood, just from a blood draw, engineer them with a recombinant retrovirus that encoded a synthetic gene that itself produced this synthetic protein that we call the CAR, and the CAR recognized CD19. And if we infused only once these T cells into mice that bore either leukemias or lymphomas, we could eradicate the cancers in those mice. And this led to gave us the impetus to proceed to clinical trials at my center. And thanks to my colleague Isabelle Rivière, who joined us from MIT, we were able to build this facility and establish processes that would allow us to manufacture peripheral blood T cells drawn from patients for infusion to those patients. I won't walk you through the early clinical studies. There were 3 teams that produced clinical results at about the same time. Ours, the National Cancer Institute, to whom we had provided our CAR molecules, and also the University of Pennsylvania that had received their CAR molecule from another group. So I'll just illustrate what these CAR T cells do from one of our earlier papers, the first one in acute lymphoblastic leukemia. And so these are patients that have a relapsed refractory disease. They've already received a number of chemotherapies, some of them a bone marrow transplant, and unfortunately, when their disease relapses and is refractory to any treatment, they only have a few months to live. And so here are subjects 4 and 5 from the first 5 that we reported on. And what you see here are leukemia cells that express CD19 in bone marrow samples a few days before receiving the CAR T cells. And you can see here 10 days or 11 days later or 2 weeks later, the complete absence, the complete eradication of any leukemic cell in the bone marrow of these patients. You couldn't detect them by sensitive methods. So these are called molecular responses, molecular complete responses. Later that year, the journal Science proclaimed cancer immunotherapy to be the scientific breakthrough of the year. That was largely for checkpoint blockade, another very successful immunotherapy, sort of an older big brother to CAR therapy. But the editors already saluted, if I can say, the emergence of this approach in 2013. The trials conducted at our center and others were soon confirmed in larger series, and by 2018, soon after the approval of these drugs by the FDA, Carl June and I summarized all this literature that clearly indicated that these results could be reproduced in the hands of many. Here are the first two CAR molecules that were approved by the FDA in 2017. Both of them are specific for CD19, that the heavy and light chains of the immunoglobulin here in the extracellular domain would bind to. Both of them have this dual signaling motif that I mentioned before. One of them uses CD28. This is the signaling domain borrowed from an important costimulatory receptor in T cells known as CD28. The other one uses the signaling domain of another costimulatory receptor known as 4-1BB, a member of the TNF receptor family. There's a lot to say in comparing these two, but suffice it to say that they achieve fairly comparable results in comparable clinical outcomes, I should say, in patients with leukemia. And so there are today 6 CAR therapies that have been approved by the US FDA. 4 of them target CD19, and 2 of them target BCMA, another target of interest in the disease multiple myeloma. And all of these CAR therapies were first tested in patients with relapsed and refractory disease and are gradually moving into earlier phase of treatment of these patients. But in those that have relapsed refractory disease, this is the complete remission rate that you obtain in different diseases. In some forms of B-cell lymphomas, such as mantle cell lymphoma, follicular lymphoma, as you see, 80% of these patients will go into a complete remission, a bit less in the case of large B-cell lymphoma, and in acute lymphoblastic leukemia, a remarkable 80% or more of these patients will go into complete remission, and about half of them will survive in the long term without requiring any further treatment, but the other half will still at some point relapse from their disease, telling us that there's still progress to be made. And so I'd like to spend the next few minutes by telling you, giving you an insight into some of the directions that we're pursuing. And so we have this foundation now, these CD19-CAR T cells that are useful in a series of B-cell malignancies, but there's room for improvement, and of course we want to expand this approach to other cancers and other diseases. I would like in the next few minutes to touch upon some directions for future work. Can we create a more sensitive T cell, and I'll explain what that means in a minute, a more discerning T cell that could tell the difference between a cancer cell and a normal cell? Can we design T cells that are both potent and durable? Often the more potent cells don't last so long, whereas those that are endowed with long-term persistence are not necessarily as potent. And lastly, mentioned briefly, can we actually bypass the need for a human donor for these T cells? And you'll see what I mean in a few minutes. So first, a few slides just to illustrate this issue of antigen sensitivity. Clinical reports taught us that sometimes patients relapse after CAR T cell therapy with the target antigen still found in the tumor. So why is it that the CAR T cell is failing to eliminate those cells? There are a number of reasons for CAR T cells not succeeding in eliminating a tumor. They may not migrate and enter into the tumor site. They may be suppressed at the tumor site. But in some cases, we know that the T cells got there and the tumor is relapsing, but with a lower level of antigen. And that led us to examine the sensitivity of these CAR T cells. And here I should bring in the physiological receptor, the T cell receptor that I mentioned earlier. It is depicted on the left here. And this is a complex that aggregates many chains, the alpha and beta chain that recognize the HLA peptide complexes, and then a variety of chains here that together initiate activation of T cells. And this receptor is known to be highly sensitive, meaning that at least under ideal conditions in vitro, a few molecules of the HLA peptide complexes are sufficient to trigger activation. But when we started looking at these CAR T cells, we found that they need about 1,000 molecules, or of course more, 1,000 molecules to initiate successful activation and then killing of their target. So that led us to create this hybrid structure that you see here, where we preserve the antibody-derived elements, the heavy and light chains, and insert them into this T-cell receptor complex by swapping out the variable regions with these antibody-derived elements. And by establishing panels of tumor cells where we control very closely the number of target molecules at the surface of those cells, we could study the sensitivity of these new receptors. So you see a natural acute lymphoblastic leukemia may have 20,000 or more CD19 molecules on its surface, but we have lines that have 2,200 or even few. The short of it is that the HIT receptor is a lot more depicted here, which is an HLA-independent T-cell receptor because it engages antigen without the need for HLA, is far more sensitive than the conventional CARs. And I can show you a few examples of that. Now, if we create an acute lymphoblastic leukemia that we know has 200 molecules of CD19 on its cell surface, the best conventional CARs we could use fail, whereas the HIT receptor will succeed in these mouse models. Likewise, in models of acute myeloid leukemia, the best CAR that we could come up with targeting a molecule called CD19 fails in the presence of that low number of target antigens, but the receptors succeed. And another example, not yet published, targets CD70 in a model of kidney cancer. And here too, in the kidney cancer, the best CARs that we can design will at best induce a stable disease. You see that when you look at the tumors in these animals, very much like what you see in patients, some of the cells stain positive for the antigen, but many others appear to be negative. In this instance, they're not negative, actually. It's just that they express very low levels of the antigen, and the HIT receptor is capable of eliminating these cells. The next topic I want to address quickly is, can we make sort of try to make a smarter T cell that is more discerning or selective in terms of its of the targets that it engages? And here I have to introduce some terminology that many of you will remember from your algebra, Boolean logic terminology. So unlike natural T cells that have one receptor that engages one HLA peptide complexes, we can of course design T cells that could engage two antigens or more. But if you think about how to engage two molecules, there are many ways to do so. You could have a T cell that recognizes antigen A or B, call that an OR gate. You could have what's called an AND gate, where a T cell would be activated only in the presence of A and B being together on the surface of T cell, but not being activated if a cell expresses either A or B alone. There are also NOT gates. We created some inhibitory receptors to antigen B. In this case, the ideal goal would be a T cell that would kill tumor cells that express A but not B, but to protect a normal cell that expresses the antigen B, triggering the inhibitory receptor. Another gate is called an if-then gate, which introduces a temporal dimension where a receptor, not a CAR, engages antigen A. This is typically the Notch receptor, which then initiates the activation, the transcription, of a CAR that engages antigen B. So in this instance, the CAR recognizing B is only expressed wherever in the body where there is antigen A. And lastly, the one I want to discuss is an if-better gate, which is, can we have a molecule B that would help recognition and killing of cells that express A when A is of limiting abundance? And let me walk you through what we're doing in the case of acute myeloid leukemia. And this is a survey of 35 patients at Sloan Kettering, looking at the level of expression of different molecules on these leukemias. One's called CLEC12A, another one AGRA2. Here's another one called CD123, and the label's missing here, but it's CD33. The Y and X axis here plot the median number of those molecules found on the surface in those patient samples. You might think that CD123 and CD33 are quite attractive because they are more abundant, and indeed there are today several trials targeting these antigens. But we were a bit fearful of doing so for the reason that's shown here. In normal bone marrow cells, these antigens are also expressed. And CD123 and 33 are found on many cells, including hematopoietic stem cells and others, which means that if your CAR T cell could not distinguish the leukemia from these normal cells, you wouldn't just eliminate the leukemia, but you would destroy the recipient's hematopoietic system. So we wanted to explore whether we could try to be more selective towards these cells while sparing the cells that express low levels of AGRA2, which we know are found on hematopoietic stem cells and what's called common myeloid progenitors. So we designed this dual system where a CAR recognizes antigen A, which is AGRA2, and this helper molecule targeting antigen B, which is CLEC12A in this case. And I'll just show you what we can do now in vivo in animal models. So on this spectrum of patient samples here, here's a cell line that expresses, you see, 10,000 molecules of AGRA2 and 500 of the helper B molecule. So we're positioned here. We put these leukemias in mice and treat them with CAR T cells that are engineered with these two receptors. And it's a low dose of T cells, but this CAR is remarkably effective and the leukemia is eliminated. You might ask, well, did the antigen B matter in this case, which was this one? No, it doesn't. In fact, if you eliminate it through genetic ablation, which means that now you have dropped your position here on the spectrum to this one here, these CAR T cells still work. And they work because there's 10,000 molecules of AGRA2 at their surface. But now, let's go lower. And now we have a leukemia variant that has 1,000 molecules of AGRA2 on its surface. So we are now positioned here. And now we treat these mice and the CAR T cells fail, which is what we wanted in this case, because we want to spare those hematopoietic stem cells. So now is the real test of the if-better gate. So now the same cell that has 1,000 molecules of AGRA2 has a high level of the second antigen B, and now these same CAR T cells work again. So this is the if-better concept. The T cell engages this antigen at this level depending on the context. In this context, it does not, and in this one, it does. Lastly, I want to, thirdly, my third example is how we can improve the functional persistence of CAR T cells. And here we have to go back to the very initial design of these receptors. The first one we reported in the early 2000s that combined this activation domain from this zeta chain from the CD3 complex and the signaling domain of this costimulatory receptor called CD28. The clinic taught us that these cells are powerful but relatively short-lived, and after a while we wondered if in fact we had created a signal that is in fact too potent, too strong, and we thought we should attenuate it and then see what happens. And so we started mutating these domains that are well known in the zeta chain that mediate T cell activation and created a family of these derivatives. And the short of it is that one of them, called 1XX, which has a reduced strength of activation, produced far better results in, again, these leukemia models. And so we brought this to a clinical trial with my colleague, again, Isabelle Rivière, and our clinician, Jay Park. We designed this trial with the following reasoning. If these cells are really more functional or for a longer period of time, meaning if they are superior T cells to what we were making before, then we ought to be able to treat patients with lower T cell doses. And I want to mention here the doses of CAR T cells that are used with the approved clinical products today. One of them, using the 41BB CAR T cells, requires the infusion of 300 million CAR T cells. Another one that uses the CD28 CAR design, which is a bit more potent, requires about 150 million of these CAR T cells. But we thought that if these cells are really better, we should be able to treat patients with the same disease, in this case, refractory diffuse large B-cell lymphomas, with lower doses. And so we started treating patients with only 25 million CAR T cells and then dose escalated. And I don't have time to go through the details, of course, so just the answer is simply that we got excellent results already at 25 million CAR T cells. More T cells didn't really make it any better. So we expanded the cohort at this low dose and found that we could get responses that match any other CAR T cell that already exists, but with a 10 times lower dose. And so this is just to exemplify. We're not going to reduce the cost of sequencing by 1,000,000 times, but at least we're reducing the T cell dose already tenfold, and I think it's going to go further than that. We are, today, things are very different from what they were just 10 years ago when a limited number of laboratories were developing these receptors. There's now really an explosion of the variations, if you like, on these CAR designs, and it's really wonderful to see. Something else that is happening is that work has inspired many to explore not just the alpha beta T cells, the conventional peripheral blood T cells that we utilized up till now, but to start engineering other cells of the immune system, macrophage, natural killer cells, even granulocytes and eosinophils. And here I want to make a comment on the sources of T cells. Everything I described to here in patients makes use of the patient's own T cells. This is autologous therapy. Some have said that it might disappear, it might be replaced by cells that you could make ready-made for infusion to patients, but that's not so easy to do. Allogeneic cells, I'll come back to that in a minute. I think autologous manufacturing is here to stay. The processes used to take 2 weeks, then 7 days, and today many protocols take 3 days, and some of them just emerging are only overnight processes. But there's a lot of interest nonetheless in using allogeneic cells from either healthy donors or cells that never were formed in the body of an individual. And that is what we might hope to achieve by using pluripotent stem cells. And you can imagine that if you have these cells growing in bioreactors, and if we mastered the differentiation process to create functional T cells, that this could actually ultimately bypass the need to go and harvest T cells from humans, including either healthy donors or patients themselves. And I have to stop very promptly. So I'm not going to highlight that there is progress in this space, but there's still a lot that we need to learn to generate authentic T cells in a dish outside of the thymus. Some of the last things I want to comment on is the expanding applications of this approach. So CD19-CAR therapy, as you can tell here, is the tip of the iceberg. There are today BCMA-targeted therapies for multiple myeloma. AML is another important leukemia for which a solution is still not found. And of course, we all await success in the variety of solid tumors. But there's already wonderful applications that are coming up in autoimmunity using these very same CD19-CAR T cells, treating patients with systemic lupus, with other encouraging results in pemphigus and other settings. In animal models, CAR T cells are starting to be used to remove, for example, fibrosis in the heart after infarction, or removing the accumulation of excessive senescent cells. My almost last comment is that really CAR T cells are disruptive in many ways. They're not conventional drugs. They're not chemicals. They're not proteins. They're actually cells. Furthermore, they are genetically engineered cells. And furthermore, they make use of synthetic genes to reprogram those cells. But the novelty and challenges that it raises extend beyond that. For the pharmaceutical industry, obviously they have to put into place a whole new manufacturing and distribution system that didn't exist until now. This is also a challenge for, reimbursements of these cell therapies that are more expensive than standard drugs. We all wrestle now with how to address the issue of equitable access, but this is going to impact the medical practice and therefore what is taught in medical schools in many ways. I just want to highlight one very crucial aspect of this, which is making these cells, producing these cells. The traditional model is that we leave that to the pharmaceutical industry. But that may not be the case for cell therapies. This field was born in academia. You might say it could just transition to the pharmaceutical industry. But I believe that there are so many diseases, some rare and some not, and so many variations on these cell types, the signaling modules, the way you produce the cells, that academia will continue to play a fundamental role in driving this field. And that gives a new mission to medical schools, one that we traditionally give and pass on to the pharmaceutical industry, and that is manufacturing. And our center demonstrates, and many others too now, that you can do that. You can build the facilities, establish teams, develop processes that empower physicians and researchers at their medical center to develop these medicines. So my final note really is that there's so much to do in the world of T-cell engineering, as you can see here. There are so many to thank as well. My mentors, at the University of Paris, at the University of Alberta, at MIT, all those that I worked with, including Anton, who's now a postdoc here in Toronto, who worked with me for the past years. There's one person I want to highlight, and that's Isabelle Riviere. You already heard her name. Because she established at our center the processes that enabled the clinical translation of our research, and without that, we would still just be publishing papers, and this would not have seen the light of day. Thank you.